family, which was named SARS-CoV-2, that causes a respiratory disease termed COVID-19. This virus has not only received widespread media coverage, but also caught the attention of scientists who have been busy analyzing the virus, the disease it causes and how it spreads. There is already an enormous amount of information available about the virus, and that is why we thought to summarize some of the information in this video. First of all, what do we know about the virus? Coronaviruses receive their name from their appearance in electron micrographs, where the pronounced spike-like protrusions create the effect of a corona, Latin for crown. The genetic material inside the coronavirus is RNA, and the entire genome is almost 30,000 bases long, packed together with a nucleoprotein into the viral particle or virion. For the virus to replicate, however, it needs to insert its RNA into cells and hijack the cellular machinery to produce new viruses. On its surface, the virion contains three main proteins: the envelope, the hemagglutinin esterase, and the spike protein. These are embedded into a lipid bilayer. The large spike protein is responsible for the crown-like protrusions, but it also recognizes the corresponding receptors on human cells called ACE2. The protease ACE2 mediates the cleavage of the spike protein, which then releases an epitope enabling the subsequent fusion of the virus with the human cell.
Researchers from China have used cryo-electron microscopy to show how SARS-CoV-2 infects humans. The study published in Science says the virus targets a type of receptor found on human cells in the lungs, heart, kidneys, and intestines. Citing Kingston University microbiologist Mark Fielder, Sky News reports that the virus seems to attack two types of lung cells: goblet cells that coat the respiratory tract with mucus, and ciliated cells that usually filter out pathogens. A previous study published in Science found the virus spike protein has two receptor binding domains, or RBDs, facing downward and another facing upward. These allow the virus to bind with and invade human cells. The virus targets a human ACE2 receptor that has bonded with an amino acid transporter. This subtype of ACE2 structure has never been discovered before. The virus uses the spike protein's up RBD to penetrate the cell. The virus then dissolves its own protein shell and releases its RNA payload inside the cell, according to British Society for Immunology. A study in Frontiers in Microbiology says a coronavirus hijacks the cell's structure to reproduce. The viral RNA takes over the host cell's endoplasmic reticulum to replicate itself and to manufacture the protein parts to make new viruses. According to the Society for Immunology, the hijacked cell's Golgi bodies then package viral RNA and proteins in a viral protein shell. This leads to the creation of new viruses that leave the infected cell via the membrane. The study in Frontiers in Microbiology says coronavirus takeover imposes stress on the host cell. Cell death or apoptosis is the result when the infection overwhelms the host cell's ability to maintain homeostasis.